Hello.
Hi.
Hello. There's Dr. Rubenstein.
Dr. Rubenstein is trying to connect.
Mm-hmm.
Here we go.
Good morning.
Good morning.
Morning.
Let's wait for 1 minute and then we can start. Is that okay if we wait for 1 more minute to have some more audience?
Of course.
All right, let's get started. Good morning, everyone. Thank you for joining today's session. Although we have worked hard to make this a smooth session, there is always a chance that some members may experience audio and or video lags or disruption. Closed captioning will be taking place during this session live. You will find the live closed captioning at the bottom of your screen. Zoom provides AI-powered live transcription that needs to be enabled by the viewers by clicking the CC button and enabling auto transcription. Now, allow me to introduce our esteemed speakers today, Dr. J. Rubenstein and Dr. Tina Stankovic. Dr. Rubenstein received BS and MS degree in engineering at Brown University in 1981 to 83. He received an MD and PhD in bioengineering at the University of Washington in 1988. He completed a neurology fellowship at the University of Iowa in 1995. And in 2004, he was the professor at Leiden University in Netherlands. He is currently Virginia Merrill Professor of Otolaryngology and Bioengineering and Director at the Hearing Research Center at University of Washington. Dr. Rubenstein has published over 110 peer-reviewed articles in both clinical and basic science journals and has mentored 18 both pre-doctoral and postdoctoral trainees in basic and translational research, as well as providing clinical training to a large number of otolaryngology residents and fellows. His laboratory studies models of signal processing in and perception with cochlear implants and is collaborating in the development of a vestibular implant. His clinical interests encompass management of tumors of the lateral skull base as well as auditory, vestibular, and facial nerve disorders. So, welcome, Dr. Rubenstein. Dr. Tina Stankovic is MD PhD and is the Bartley Foundation Professor and Chair of the Department of Otolaryngology, Head and Neck Surgery at Stanford University School of Medicine. She is a Harvard-trained ear and skull-based surgeon and a Massachusetts Institute of Technology-trained auditory neuroscientist. Her clinical focus is in otology and neurotology, and she previously served as chief of the Division of Otology and Neurotology at Massey and Ear, a teaching hospital at Harvard Medical School. In addition to her clinical and administrative duties, she directs a basic science laboratory focused on improving diagnostics and therapeutics for sensorineural hearing loss and vestibular schwannoma. She has initiated and led successful cross-departmental national and international collaborations to develop and deploy novel molecular diagnostics and therapeutics for hearing loss and otologic diseases in general, while educating the next generation of leaders in surgery and science. So, welcome, Dr. Stankovic.
Thank you.
So now I would like to ask our esteemed speakers to share their experience. How did they become interested in clinical research, clinics and research areas of their focus, and how did they pursue their career so far? Dr. Rubenstein?
A broad question. I was originally not interested in clinical medicine at all. I was in college. I planned to be an engineer, specifically working on signal processing for musical instruments was my big interest, and was working in a laboratory where I built hardware for visual physiology research. And in the process of trying to build better hardware, I observed some of the experiments, which were Hubel and Weasel era visual cortex single unit recording experiments in cats. And that kind of got me interested in neuroscience. Neuroscience got me interested in hearing, and hearing got me interested in cochlear implants. And cochlear implants got me interested in becoming a neurotologist. So I became a clinician through the process of becoming a scientist, and it's always been a key part of what I do. So to me, they're not sort of. Well, let me put it this way: when when I was in the MD PhD program at the University of Washington, we had Nelson Kang as a guest speaker. This was in probably 1987. And he gave a long lecture about how there was a very different culture between clinicians and scientists, and that it would be very challenging or impossible to bridge the two effectively. I became very close with Nelson as of that date because I decided it was my job to try and prove him wrong, and proving Nelson wrong was always a challenge. So that interaction sort of really sort of clinched it for me because I realized that medicine provided opportunities for research that I would not necessarily have as a scientist without clinical training. And science provided me opportunities as a clinician that I wouldn't have without a real scientific background. So that kind of sums up what my experience was, and the key to the other key to my experience, as I'm sure Tina will describe for you as well, is having been an MSTP student. MSTP is really the only environment in the current medical enterprise which really pushes young people to become both clinicians and scientists. And one needs really good mentors to push you in that direction if one doesn't have a program like that that's already moving you that way.
Thank you. It was a great journey and lots of experience and different backgrounds. And Dr. Stankovic, you want to share your experience?
Absolutely. My interest in bridging basic science and clinical medicine dates back to my undergraduate years. I was a double major in physics and molecular biology, and I was looking for a project that would allow me to bridge the two along with my budding interest in medicine. And this is where I discovered hearing and worked in a cochlear implant lab with Don Eddington and Jay Rubenstein, modeling how the auditory nerve responds to electrical stimulation. And that was a very formative experience because I got to interact with some patients as well and noticed that there is a broad spectrum of their responses. Back then, some were using cochlear implant primarily as a lip reading assisting device. And that stimulated my interest in understanding how the brain controls the function of the inner ear, which led to a PhD studying in particular that question, which I did in parallel with my MD training. So the two, in my case, just like in Jay's, have been very intertwined and have reinforced each other all along the way. And I found it to be an incredibly fulfilling and exciting endeavor.
Sounds good. Thank you so much for sharing your experience. So, what does your typical week looks like as a clinician scientist?
Are we going to continue in this order? Do I go first?
Mm-hmm.
Okay. I'm not sure I have a typical week. One of the things required to be effective in both domains is to be very flexible and use time as a resource and apply time to whatever problem needs to be solved at that time. I do about 50% clinical work and 50% research and administration, and it varies a lot. I have one day where I reliably operate all day, I have one day where I reliably see patients all day, and then the 3 other days of the week are highly variable. I could be reviewing, I could be writing, I could be mentoring, I could be trying to fix something in the lab. It's quite variable.
Okay, Dr. Stankovic.
I completely agree with that attitude and the need for flexibility and the need to seize opportunities that present themselves, such as new research collaborations, or early on, as you are establishing yourself as a surgeon, also new opportunities to pursue some complex surgical cases as well. So my time split likewise varies, but what is fixed is that I also have 1 full day in the operating room per week and 1 full day in clinic per week. And the rest of the week is variable. And that has also evolved as my responsibilities have changed. Before I became department chair, then there was even more flexibility how I spent those 3 days addressing research and administration. And now there are more meetings that I'm required to attend. And it's actually really fascinating. And I love learning. I think that's common to all clinician scientists. We are curious by nature. We like solving problems. And there is certainly lots of opportunities to do that being a department chair as well.
So, like both of you are like professor or department chair on such positions, but like for what is your advice on someone who is starting their career as a clinician scientist and is a new faculty, how to devote time and how to distribute time and their responsibilities within a week or within a month? Like, how can they efficiently organize their time and also have work-life balance?
I'm the wrong person to ask about work-life balance. That's not something I've ever been. That's never been my strong suit. Let's put it that way.
Mhm.
Because to do clinical work and scientific work at a high level requires a lot of time, and it requires more time than goes into a standard work week.
Yes.
And it requires, well, I'll back up. The first question before a junior faculty member is trying to divide their time is, where did they decide to take a job? Do they work at a program that supports clinician scientists? Because you have to have colleagues that support what you're doing. You have to have a department chair who supports what you're doing. I highly suggest working for Tina. I can't emphasize enough how important that is. And even beyond your department chair and your colleagues, are you at an institution that values what clinician scientists do? There are a number of medical school deans who think, clinicians should do clinical work and bring in clinical money, and scientists should do research and bring in NIH grants. And they're not interested in people trying to do both, because they like Nelson in 1987, they don't believe that can be done effectively. So one needs to be at a supportive institution and in a supportive department. And then one needs to choose, Tina and I are both neurotologists, but clinician scientists in otolaryngology are not, uniquely neurotologists. They can be in a variety of other clinical subspecialties. And so the specifics will vary, because the type of procedures you do as a neurotologist are different than the type of procedures you might do as a head and neck surgeon who has a research lab doing molecular biology of cancer, for example. And one needs to look at the areas of science that you do and the areas of clinical work that you do, and map out a pathway that works. For me, that was to choose a clinical area and a research area that closely overlapped. So if I'm reading clinical papers in my clinical field, they commonly overlap with basic science I might read in my research domain. And you need to find efficiencies of overlap that help you. And for me, that was the pathway. But I'm sure there are many other pathways that people can choose. But you do have to give some thought to how can I efficiently do both of these things, and how can I take advantage of their overlap to optimize my time.
Great.
Yeah, these are really wise words, and I absolutely second them. In addition to what Jay has said, I would say that even before you become a junior faculty member, during your training, it's really important to learn the material exceptionally well, because once you start your clinical practice, you will not be spending as much time seeing patients or being in the operating room as someone else who is doing it full time. Likewise, in research, you will not be doing it 100% of the time because you'll be splitting your time. So it's really important that you become skilled and comfortable in thinking independently. So during your PhD work, really embrace every opportunity to dig deep and learn how your equipment works. If coding is an important part of what you do, learn to be skilled in that. Establish collaborations with people that you can build upon. And in the clinical realm, you really have to learn the material, because you don't want to get a label that you are a half clinician and a half scientist in terms of your skill set. And in fact, if you've acquired the skills that you need, that allows you to pursue exciting and impactful questions that, as clinician scientists, we should be doing, because we have the ability to not only identify problems that need to be solved, but actually help solve them. And so now, when you're done with your training as a junior faculty, it's absolutely essential to have the environment that's supportive, which we already heard from Jay about, to have the infrastructure in place. And that infrastructure consists of people, primarily, people who understand you, who are supportive of your endeavors, but also the environment. Do you have equipment that you need? Do you have access to core facilities? And is that all accessible? Because if you're on completely different campuses when you're doing clinical work versus research, it can be tricky. It's easier if it's close by. And then in terms of work-life balance, I like to think about work-life integration instead, because balance implies that somehow you put them on a scale and they have to be evened out. And integration really means that you're living a full life and you are integrating everything that you have learned, and it becomes a part of your identity. So indeed, lots of problems that we work on require a lot of time, and that time can occur even if you are taking a walk with your children, or you are on vacation enjoying the beauties of whatever, the Hawaiian Islands, and still an idea comes to your mind, and I would not ignore it because I'm now on vacation. In fact, I would embrace it because it's an opportunity to think about it in a completely different context, free of daily obligations that you have to attend to when you are formally at work. So indeed, it is work-life integration and enjoying your life, because who is there to judge what you should and shouldn't be doing? As long as you are enjoying the ride and people around you are enjoying your company, and that includes your family, that's all that matters.
That was quite enlightening. Thank you, Dr. Stankovic. And there is a follow-up question about like how have your experience in the clinic informed your research questions? Like how is it helpful to design your research questions based on your clinical experience?
So, my first experience interacting with patients was doing psychophysical experiments in people with cochlear implants, because that's the first human subject research I did in my career. Prior to that, I spent a lot of time with cats. And it became really clear, back in that era, cochlear implants couldn't do very much from a clinical standpoint, but they could clearly make people hear sounds. And at that time, we were really trying to figure out what do we need to do to the signals so that the sounds make more sense. And it became really clear to me very early on that listening to what the patient said and how they described their sensory experience was going to provide me information that I could obviously never get from single unit recording in the auditory nerve. Which isn't to say that I didn't go on and do single unit recording in the auditory nerve also. But it made it clear that I could get information from patients that I couldn't get any other way. And likewise, descriptions of other, I've done some research on tinnitus and some research on vestibular disorders, and it was really my clinical experience in those areas that drove me to try to study interventions for those problems. From being aware about how common those problems were and how distressing they were for many, but also features of them that I thought perhaps I could explain based on scientific principles that I understood about how the auditory system worked. That really is, kind of summarizes my experience in that area.
Thank you.
And all of the research that we do in my lab is inspired by clinical problems, unsolved clinical problems. So there is really a very close integration between the two. We use a variety of different techniques to address these unsolved medical problems. And we are very focused on sensory neural hearing loss. So in particular, the problems that we study are the problems that patients ask us all the time, such as, what exactly is wrong in my inner ear? Why do I have sensory neural hearing loss? And today, technology doesn't exist to answer that question precisely, because even if we establish, let's say, a genetic cause of hearing loss, we still don't know what that inner ear looks like. And that question that I was hearing from my patients repeatedly stimulated a research direction where we are developing a tiny endoscope that we can insert into the inner ear to see microanatomy and hopefully answer these questions for living people. Because today, the pretty much only source of information about what happens in the human inner ear at a cellular level comes from studying autopsy specimens, but that's not that relevant for living people. So that is one of the questions that has been stimulated by clinical problems. Another question that patients ask is, well, can you biopsy my ear? Because I had a biopsy of my gut, or I had a biopsy of my skin, and this is how they told me what I had. And today, we cannot really biopsy the inner ear because it's such a tiny organ that tissue biopsy would destroy it. However, that stimulated our direction in performing liquid biopsy of the inner ear. And that is feasible. There's some fluid in the inner ear. In the human inner ear, it's about 150 microliters. So even if we take a little fraction of that, we can detect signatures that are different in people who have normal hearing versus those who have different types of hearing loss. So we envision a future in which liquid biopsy of the inner ear may become routine if done safely. Another question that has inspired our research is when we see patients with these tumors in their head, they're called vestibular schwannomas or acoustic neuromas, and some of them have very tiny tumors and a lot of hearing loss. They can be profoundly deaf, and others have large tumors and no hearing loss. And that begs the question of how do these tumors cause hearing loss? And textbooks say it's by compressing the nearby auditory nerve, but it really doesn't explain what we see in clinic. So for us, that led to this new direction of understanding the secretome of these tumors. What do they secrete? And can these secretions affect inner ear function? And the answer is yes. And we have identified these secreted molecules even in patients' blood, and they correlate with some of the clinical findings. So these are just a few examples, but the big picture conclusion is that clinical medicine is a huge source of inspiration for everything we do.
Great. So my next question was about, like, how is the approach of a basic researcher and a clinical researcher different or could be similar based on their diverse different backgrounds, and where do they actually start converging? Like, what is your take on that?
Yeah, so I'll go back to Nelson Kang. The cultures, at least historically, were extremely different, and that's changed over time as we have a lot more clinician scientists working now than when I was in training, of both, and we have a lot more PhD scientists working in clinical environments than we did when I was in training. So there's a lot more integration between science and clinical practice than there used to be, which I think is great for all involved. But they are different, and when you do a PhD, you take for granted certain things that you've learned. And one of those things is that whatever problem you're thinking about, somebody has thought about it before. And so when you're attacking a new problem, the first thing that you do is figure out who's thought about this before, who's worked on this before, what techniques did they use, et cetera, et cetera, et cetera. That thought process does not come naturally to an otolaryngology resident who does not have an extensive research background. Both Tina and I are in departments that have training grants to support basic science training for otolaryngology residents. And one of the common problems that I see in my residents is that when they think about something, they think they're the first person that's come up with it. And they fail to adequately review literature on what's been done before in this area, which is not something any graduate student would ever do, because they've grown up in an environment where that's the first thing that you do is look at what's been done before. The different, but what an otolaryngology resident might do that a graduate student might not do is be more willing to tackle problems that require or that have a lot more uncertainty associated with them. In medicine, you get really used to making decisions with partial information, for better or for worse. We are forced to make decisions every day, many times, with only partial information. And so a PhD student is unlikely to do that because that's completely contrary to the scientific method. You get all the information first, and then you take the next step. But in clinical training, you're forced to take the next step without complete information, and this for many is a challenge. For the residents, it's a challenge to they write proposals where they clearly haven't reviewed the existing literature in an area because they didn't know it existed because they didn't look for it, and for PhD students, it's a level of caution that if you're trying to do clinical research is challenging because, as Tina said, we can't, get a soft tissue biopsy of the cochlea to figure out, why this patient has sudden sensory neural hearing loss.
Yeah. Dr. Stankovic.
Yes, very insightful, Jay. In general, there are actually lots of similarities between basic researchers and clinical researchers in terms of how we tackle problems if we assume that clinician researchers have had scientific training. I think what Jay has highlighted really nicely is the issue can arise if people have not had the training. But if you have had the training, then your approach is similar in that you dig deeply, you use whatever technique you need to solve the problem. I think the main difference may be that basic scientists are sometimes entirely curiosity-driven, which is very exciting. There isn't necessarily a practical end to it. I think lots of clinician scientists ultimately are interested in a practical application or implication of what they do, whether that's a new device or a new prognostic or a new way of thinking about how to evaluate or treat patients. But on the basic science side, it's not necessary that that be the end of the endeavor. And however, it could turn into an amazing breakthrough. And in fact, lots of basic science works that way. And that's why it's so important to continue to support basic science in clinical departments. And a great example is Jennifer Doudna, who won Nobel Prize for her research on discovering gene editing, and that led to that whole CRISPR revolution. But when she was interviewed about her work, before she really recognized the incredible impact this could and will and has had on how we consider practical things, including in medicine, she was commenting that it sometimes feels that she's working on such esoteric things that it's not clear whether any of it will make a big impact. But she continued to work on it because she was entirely curiosity-driven. And that curiosity-driven research is critical to make new advances, or how Horowitz discovered, he won a Nobel Prize for his discovery of apoptosis. And he was working on a seemingly very tedious problem. He was counting cells in a worm and noticed that at a certain age, the cell number changed. And that observation, which was a very astute observation, led him to ask the question of why. How does that happen? Is there a program to it? Why do these cells disappear or numbers change at a very particular time point? So I think both approaches are really important, and the collaboration and integration of these approaches is so important. Collaboration between basic scientists and translational scientists and clinician scientists and clinical scientists is really important. And departments that have all of that are really lucky, because it's in that very fertile ground where people have slightly different perspectives that you are really allowed to flourish in terms of out-of-the-box thinking. And out-of-the-box thinking is critical for solving unsolved problems that have been around for centuries and even millennia.
Thank you so much. We have a question from audience, Bryce. He said, for medical students earlier on in their career, how can you get clinician scientist training without the opportunity to do MD or PhD?
Yeah, there are a lot of opportunities, both within medical schools and outside your medical school, to get funding to spend a year in a laboratory. So, for example, in our T32 and upcoming R25 program, we have a slot for a medical student to take a year off from their clinical training to get basic science training. And there are many, many other opportunities like that. There are foundations that support that. It's one of the wonderful things about wanting to become a clinician scientist is there's a lot of financial support out there to push you in that direction, because there is a perception that there are not nearly enough clinicians that have basic science training. And I would encourage you to look into those opportunities, because nothing teaches you to be a scientist like dedicated research time with a good mentor.
Absolutely, absolutely. And indeed, there are opportunities to do that in our department. We have multiple very successful, top-notch clinician scientists who do not have a PhD degree. They have MD degree, and they did precisely that. Took a year or two of dedicated research in a lab. And there are numerous funding opportunities. Just to put in a plug right now, we have an opportunity to apply at Stanford for a clinician scientist training program year. And currently we have one of the medical students from Duke who came to Stanford to spend a year with us, and many institutions, the University of Washington, where Jay is, and many other institutions across the country who are invested in training have such opportunities. NIH has such an opportunity as well. It's a great program where you can spend a year in the NIH labs, also affiliated with the NIH hospital, which is a major research hospital in the country. So that is the way. It's really spending the time to learn the way of thinking, to learn about scientific rigor, and to learn new techniques so that you are equipped to answer questions that you become interested in solving as you continue your journey with patients.
Thank you. So I have a follow-up question, but it's like just opposite of what was the previous question. Like, do clinician scientists have some like training program for basic research scientists? Like they can have an idea like how does the clinic work and like what is their mode of working and dealing with patients, and how do they recognize problem, for example, hearing loss in the patients and all that, so that they can like build on and try to find or know, try to pursue a new research question.
I'm not sure I completely understand the question.
So basically, like, is there any program or training program, or like if basic research scientists like who are not clinician and they want to understand about, know basics about the clinics in hearing research?
Yeah. So I don't know of specific programs that are oriented that way. You know, in our department, we commonly have visitors observing in the clinical environment. Um, Typically, it's not established scientists, it's students, but they are PhD students who are interested in experiencing what a clinical environment is like, either because they're interested in studying clinical problems or because they're ultimately interested in becoming clinicians themselves. You know, in the same way that, you know, in response to the earlier question, I said nothing teaches you to do research like dedicated research time with a really good mentor in a basic science laboratory. You know, nothing teaches you about clinical problems like being in a clinical environment. And that could mean observing interactions in patient interactions in a clinic. It could involve being in the operating room and observing how procedures are done. You know, we do a lot of intraoperative electrophysiology, for example, and that's a great opportunity for an electrophysiologist to understand what, you know, how electrophysiology is applied in a clinical environment. So yeah, I think there's not a lot of organized programs like that, but I have to correct myself. Our bioengineering graduate program actually has a program like that built into the graduate program, where the students can request dedicated time to observe in a variety of clinical environments that they're interested in. Outside of that, I don't know of any other organized programs.
I do. I was a part of that program for a while when I was still in Boston. It's the Speech and Hearing Bioscience and Technology PhD program. It's NIH funded program, and an essential component of the program is clinical training. I used to direct that course, which is called Clinical Aspects of Speech and Hearing. And the whole idea was to expose PhD students to major problems in the field of otolaryngology. So the expectation was not for PhD students to become conversant in medical terminology fully. However, they started to understand what the problems are. What are the diseases that we see in clinic? What is Meniere's disease, and why is that still an issue? Or what is vestibular schwannoma or acoustic neuroma? And what are the issues that patients experience? And for PhD students who have gone through that program, that has been a formative part of their experience. They take anatomy lab where they actually dissect a cadaver. That's an incredible bonding experience for the whole team. And it allows PhD students to establish some close links with clinicians, which then can evolve into long-term collaboration, so that PhD students, after they finish and now they are independent investigators, they continue to have a source of clinical inspiration through ties and connections that they've made through their training program with their peers, because they observe in clinics where there are medical students and residents, but also with their mentors. So that's one of the programs that comes to mind. Another one is actually here at Stanford, there is a new program in medical physics. And that's through the combined department of radiation oncology and radiology. And these are PhD students, but they are fully immersed in clinical programs, and they take both basic science courses and observe in clinics. and in different clinical settings. We have in our department here a course on that's designed for PhD students, a course on how the auditory system works. And as a part of that course, students have an option of observing in clinics and operating rooms. So there are certainly opportunities, and if someone knows that this is something they're interested in, there can be customized programs, because clinicians in general are very eager to have people shadow them, accompany them in clinics and operating rooms. And the clinician in general like to teach, because the profession is so dependent on the transfer of knowledge that we're talking all the time. We are educating patients when we talk to them in clinics, and in the operating room, we are teaching our trainees how to do surgery. So we are talking all the time in terms of what steps to do and what the challenges are, and that can be a great spark of ideas for PhD students.
So whenever a clinician scientist want to collaborate with the basic research scientist, so what do you majorly focus on? Like, is it like just like you want to do something like which is not feasible in your lab, but someone in the basic science has that facility, or other things are also taken into consideration despite expertise and the facility?
I would say that's not fundamentally different from if you're a basic scientist who needs to collaborate with another basic scientist because they have skills and technologies accessible to them that you do not readily have access to, and you have something to bring to the table as well. I don't think that's a fundamental difference between, a clinician scientist collaborating with a basic scientist. I don't think that's fundamentally different from two basic scientists collaborating with each other.
I completely agree. Yes, it's organic, and it depends on complementary expertise that's required to solve a problem.
Yeah. So I would encourage our audience to ask questions. Please don't be shy, and please try to write in the chat box, or you can just ask the question directly to our speakers. So there are like multiple open questions in hearing research. How do you like pinpoint to pursue one particular question or to pursue one particular area of research?
Okay. I would answer that with two things: interest and opportunity. Is it a problem that interests me? And I'm not sure I can define how a problem has characteristics that makes it interest me, but it has to be interesting to me, and I have to have the opportunity to study it. And that sometimes comes out of the blue. And very commonly from trainees who bring novel questions and novel ways of looking at old questions to your attention. So I just published a paper in hearing research on the psychophysical characterization of human subjects with STRC mutations. Not something I ever would have thought I would have done, 5 years ago, but an opportunity arose when an MD PhD student working in a well-known genome scientist laboratory came to me telling me they wanted to characterize hearing loss genes, and they proceeded to do that. And then they came back and said, well, I'm interested in learning more about the phenotypic characteristics of these different hearing losses. And I said, well, what are the most common genes that you've encountered? They particularly were interested in cochlear implant outcomes, and they didn't have enough patients to really study that effectively. But a very common mutation that they identified was stereoselin deficiency. I asked him, well, what does that actually do? And he described it to the best of his ability at the time. And then both of us researched it further. And what we found was that it basically disconnects the cochlear amplifier. It separates the outer hair cells from the tectorial membrane because the structural protein that normally connects them is missing. And I said, well, what a cool opportunity to look at the behavioral effects of the cochlear amplifier in human subjects by looking at people who don't have one and have otherwise completely normal ears. So he and I and several other people then went about, characterizing what the behavioral deficits were in these individuals, and so that was interest and opportunity. My lab has a lot of tools for studying psychophysics in cochlear implant users, and we've also studied a lot of people with normal hearing to compare to. And here we had a population of patients who we've defined to have this particular mutation, and we have a facility that allows us to do behavioral studies in human subjects, and those things combined to create an opportunity to study something that I never would have imagined studying before, but it was very compelling to do because of my interest in detailed specifics of hearing loss, and the opportunity provided by this new patient population.
That's such a beautiful example, Jay. And indeed, in addition to this selection based on interest and opportunity, I would add the so what question. It's worth thinking about what is the potential impact of what you're working on, because if you have so many things to choose from and now you have to pick, and even after maybe more of them interest you than you have time to pursue, so then you can stratify them by answering the so what question. Because if you solve that problem, will it impact the field or patients in a major way? And I can tell you that that's how I pursue this question of developing an imaging probe to see inside, hopefully, a living human inner ear. When I first approached an optical engineer about it more than 10 years ago, he asked me, well, who else is working on this? And back then, it was really a problem that nobody was working on. And he said, well, it must mean one of two things. It's either incredibly difficult or it's completely irrelevant. And I said, well, I can assure you it's not the latter. It's highly relevant, but it's an incredibly challenging problem. And he then became intrigued. But it also took time to get him more interested, because he also didn't want to work on something that was so difficult that it would take forever to make an impact. But it took a really interested and inquisitive graduate student who was willing to come visit me in my lab and actually spend some time looking at human samples, and thought, oh, this is actually a really cool problem. I can see how I could apply my training in physics and optics to solve the problem. And now, more than 10 years later, we have, and with a different collaborator along the way, we now have a prototype device that we are testing in human cadavers, and are very hopeful that we'll do first in human testing within the next year or two. But it's a problem that I started working on because of the potentially big impact, the so what question, knowing that it may not work at all, that it may fail miserably because it's such a difficult problem. But even if it failed, I would still feel fine about that because I would know that I have spent time working on something that is important. And even if we don't solve the problem, we'll provide some building blocks that will help someone else solve it. So that's where that first comment from Jay is so highly relevant. Is it interesting to you? Is it fulfilling to you?
I'll follow up to what Tina said. The so what is a critically important piece, obviously, if you're trying to get the work funded. That's sort of the other component that we haven't spoken about here. But the so what, it's got to be interesting. You have to have the opportunity. But if the so what is not really, is not really big, it's going to be hard to get the work funded.
Yeah. We have another question. Is the research or skills you did in your training similar to your current work? If not, how did you learn new skills while having a busy clinical practice?
In my case, my graduate training was in neurophysiology and computational simulation. My postdoctoral work is where I first learned how to do psychoacoustics. I continued doing modeling for a long period of time after that and still do it today. But I only did a 1-year postdoctoral research experience, and I didn't really learn enough psychoacoustics in that period of time. Going to lots and lots of CIAP meetings and lots of ARO meetings didn't completely repair that. And yes, that's a very important question. It's very hard to learn new techniques when you have a busy clinical practice. And in my case, I was fortunate enough to be awarded a sabbatical where I had funding both from my university, which was the University of Iowa at the time, and from the university I spent time in at the University of Leiden. I had a dedicated 9-month period to brush up my psychoacoustic skills so I could work more effectively in that environment. That was a really sort of key part of my sort of scientific journey. Was I didn't learn, it's not that I learned new techniques, it's that I became more skilled at using the techniques that I had first acquired during my postdoctoral experience.
Yeah, I think it's really important to acquire skills and keep acquiring new tools along the way because science evolves. And I can tell you that my PhD in terms of techniques that I relied on included systems electrophysiology and single cell recordings from the auditory nerve. And then for my formal postdoctoral training, where I took 2 years during my residency to really dedicate that time to learn a new set of skills, I learned more about molecular neuroscience and transgenic mouse models of all sorts of diseases. And then when I went back to finish my clinical training, I still had some time in the evenings and weekends, and I was eager to acquire additional skill sets. And I learned about bioinformatics. A lot of it was self-taught because I got books that I read, and then you could take online tutorials through the Broad Institute at Harvard and MIT, and those were really great courses. And that allowed me to actually analyze data even when I didn't have the time to perform experiments myself. And then in my independent lab, I continue to build on these building blocks, just like Jay. That's really critical because that's the foundation that you have, while continuing to expand our tool set. Because, for example, gene editing that I mentioned didn't really exist when I was a graduate student, and I had to learn about it. What does it really entail? How does it work? Can we use it as a tool? And now we use it as a tool in the lab because it's so helpful. You can very quickly generate different cell lines that have phenotypes that you are interested in studying. Another major new direction for my lab, which is more recent, was expansion into the stem cell field. And initially, I was reluctant because I'm not a stem cell biologist. But I decided that this was so critically important because, again, when dealing with patients, it was incredibly frustrating telling them over and over that, yeah, all these drugs work in mice for restoration of hearing, but none of them work in humans. We still don't have any drugs or biologics that are relevant for human hearing restoration. But that's a big problem, not only for the hearing field, it's for all of medicine. 90% of clinical trials fail. That's a mind-boggling statistic because these drugs and interventions that make it into clinical trials were selected based on their success in animal models. So now if 90% of them fail, that's a very labor-intensive and an incredibly expensive failure because it can take 10 years to figure out that it's a failure. And it takes more than $1 billion to figure it out. And that's why I said, wow, well, we should be testing some of these drugs on human cells, but we can't get hold of human inner ear cells because we talked about the fact that we cannot biopsy the tissue. But in the meantime, this new technology became available, which is induced pluripotent stem cells. Yamanaka won Nobel Prize for figuring out how to turn any cell in the body into a stem cell. So now that we can make these stem cells from patients, we can then convert these cells, differentiate them into inner ear cells. And again, for me, that was a major shift in the lab. I hired a couple of postdoctoral fellows who had their PhD degrees in stem cell biology. I established collaborations with leaders in stem cell biology field. I started attending their meetings and journal clubs. And now it's actually a very exciting and vibrant area of research in my lab, and it complements everything else that's happening. And people in the lab collaborate with each other because we have electrophysiologists in the lab and modelers and molecular biologists and immunologists and stem cell biologists. And all of them are working side by side to address these unmet medical needs. So the bottom line is that I think it's really important to learn skills really well during your training period, because these are the skills that you will continue to build on and refine. And in addition, it's important to continue to expand your toolbox because science keeps changing and new tools become available.
I have a follow-up question about patient-specific human stem cells, where you talked about a direct conversion of these cells into hair cell-like cells or other cell types. Like how challenging it was, or is it like easy to do in a dish, or there you had to do some modifications in the protocols to convert them into hair cell-like cells or other cell types of the inner ear?
It's not straightforward because that actually plagues the entire stem cell field, the issue of robustness and reproducibility. Because sometimes even if you use the same protocol, the same recipe in two different labs that are on the same floor, you'll get different results. Even if two different people in the same lab attempt to use the same protocol, they'll get different results. That's incredibly frustrating. And that is a big deal. That actually, again, plagues the entire stem cell field. So we have been working on addressing these inconsistencies and trying to solve them. And it's very tedious work because it takes, it's permutational. It takes altering things one at a time, documenting them really well, and then redoing it to see whether now if you follow your own recipe really precisely, will you get the same results? And if you don't, then what happened? Why not? What are the lessons we can learn from experiments that give us different answers? Is it because of our technique, or is it telling us something about basic biology of cell differentiation? But a comment that I'll make along these lines, changing direction like that can actually be very challenging in terms of funding. Because when I initially applied for grants in this field, then reviewers were really harsh, saying, well, that's not your field. And it's also very frustrating because you can say, well, I've established myself as an investigator who can solve problems, and I can acquire tools that I need along the way and look at our track record of what we have done. And still reviewers would say, yeah, but this is a different field, and it's really challenging. And this is where we go back to having the infrastructure and the environment that supports you and appreciates what you do. We've been really lucky to have had philanthropic support for that kind of research to get it off ground. We still don't have governmental funding for that research. We have a couple of grants pending, so we'll see what reviewers say this time around. But basically, we had to get papers published to establish that this is something that we can do. And that goes back to the whole funding scheme and having the environment at your home institution that supports you and appreciates the new direction that you may be formulating, and that may take a while for the scientific community at large to recognize so that you can get funded.
I'd also point out that although I've never used one, NIH does have mechanisms for mid-career scientists to train themselves in new areas and get actually mentorship from somebody where it is that field. And if I were in mid-career and trying to totally change direction, I would strongly look into one of those opportunities to fund that, because the whole purpose of those mechanisms is to allow mid-career scientists to learn new techniques that are beyond what they learned in their original training. I'll also comment that, going back to Bryce's first question, a great example of a clinician scientist who does not have a PhD is Richard Smith at the University of Iowa. And Richard, as far as I know, is the only otolaryngologist to ever simultaneously hold 3 R01s. And one of the decisions that he made when he got his 3rd R01 is he was actually going to scale back his clinical practice, because at some point it becomes, as much of an enthusiast of clinician scientist as I am, at some point it becomes non-tenable to try to maintain a busy clinical practice and run that magnitude of research enterprise. More commonly, we see the opposite. We see clinician scientists who decrease their scientific activities and increase their clinical activities. But there's no rules about this. One Richard was really interested in the research that he was doing, and that pushed him to become more of a scientist and less of a clinician, at least in terms of the hours per week that he spent in these activities.
Thank you. So
Yes,
sorry.
That comment on busy clinical practice, I think the key is preventing it from being too busy. And you heard from both Jay and I that we have limited our clinical time, and it's intentional, it's by design. Both of us have one day in the operating room, one day in clinic. Sometimes there can be more time in the operating room depending on what you see in clinic. And you don't know that because people come to you with whatever problem they have. But neither of us started with 2 days in clinic and 2 days in the operating room, because if you do that, then your clinical practice is indeed so busy that you don't have time to do research.
So what is your major piece of advice for future clinician scientists for what to do, what not to do to become successful in their career?
Mentors and environment are the two words that come to mind with that. And also just being confident that this is the path you want to take. As in our discussion of work-life balance, to do both of these things well requires a pretty hefty commitment, and one has to think carefully about whether one wants to commit like that. But if the answer is a definitive yes, then the next question is mentors and environment. And I say mentors because there's not going to be one mentor for you. There are going to be multiple mentors for you. And you want to surround yourself with the best possible mentors in both clinical and basic science domains.
And I would add to that that it's really important to follow your passion and not to compare yourself. As human beings, it's natural that we like to see where we stand compared to our peers. And a little bit of that is fine, but if you overdo it, it's such an energy vampire that prevents you from doing what you are here to do. And it's important to live your life, not somebody else's life. People may have expectations on you, whether it's your parents, your partner, your siblings, your culture, your society. They may have all these expectations of what you should be doing given what you have done thus far. It's fine to consider it all and then live your life. What is it that makes you tick? And each of us is here on this planet with a unique purpose. All of us are here with our unique sets of skills. And it's important to contribute in that way. And if you're spending a lot of time comparing yourself, then you're wasting a lot of your precious time to do what you are here to do.
Great. So one last question I would like to ask is how challenging and different it was to work during the pandemic for you guys as a clinician scientist.
So, for a significant chunk of time, we were not allowed to do human subject research at all. And that shut down a big chunk of what I do research-wise. The clinical activities were only shut down for a significantly shorter period of time. So it impacted both clinical and research activities, but it had a much bigger impact, or a longer impact, I should say, on research activities. I have a close colleague who was unable to get the data that his next grant renewal depended on as a result of the pandemic. So that whole area of work in the center that I run is gone. because he was not able to take the next step due to the impact of the pandemic on access to human subjects. Obviously, it had lots of other impacts throughout all kinds of research, but again, it was a much bigger research impact than it was a clinical impact. I'll stop there because that kind of summarizes my thoughts about the question.
For me, the pandemic really reinforced a deep sense of gratitude for the incredibly fulfilling career that I've chosen. And what I mean by that is, as clinicians, we went back to work pretty much right away. And as social creatures, being surrounded by people is just so fulfilling. I noticed it in my own children when they had remote schooling and were isolated at home, and Boston was the epicenter in the United States at the time. I really noticed how it affected them. And I felt lucky that I could go to work in person and be surrounded by living people in 3D as opposed to 2D alone. And you could feel how lonely our patients were and how grateful they were to be able to see another human being in 3D. And our patients with hearing loss in particular were so deeply impacted because all of a sudden everybody was wearing masks and they already have issues understanding people, and now they cannot understand anything anymore. And that led to even more isolation, social isolation, more depression. So being in a situation like that, being able to reassure someone or listen to them carefully, because we were not seeing as many patients at the time, we actually had the luxury of time, the luxury of listening, was so incredibly fulfilling and bonding that I thought, wow, I feel really lucky to be a clinician. And then as a scientist, I also was really lucky because fortuitously our new change in direction, actually an expansion of our research direction now included stem cells. And at that time, we were really new in the stem cell work, but then the pandemic occurred. So we said, okay, we're starting to see patients in clinic who have hearing loss and dizziness in the setting of COVID. Could they be causally related? And could the virus, SARS-CoV-2 virus, directly infect the human inner ear? And we could actually continue our research because we changed our direction a little bit to now start studying COVID and its effect on audiovestibular function. And pretty much the only type of research that was allowed in basic science labs at the time was COVID-related research. And we had our stem cell biologists who came to lab every day of the pandemic. And it was a collaboration with a lab at MIT. And to make the long story short, we actually were the first to discover that SARS-CoV-2 can directly infect human inner ear cells. And we used stem cell-derived inner ear organoids and otic prosensory cells and Schwann cells. And that stimulated our ongoing research in the stem cell field. So for me, that was a silver lining of the pandemic that I really would never have anticipated in terms of how it furthered my appreciation for the entire field, the field of medicine and medical research and being a clinician scientist, and then actually turning an ugly situation into an opportunity to discover new things and impact patient care.
Tina has just taught us how to turn lemons into lemonade. It's that opportunity thing that we were discussing earlier, and she had the wisdom to see the opportunity present in a situation that pretty negatively impacted many areas of science.
So is there anything you want to include, or the question I missed, but you want to talk about before we conclude the session? Because we are heading close to the time.
Not for me, other than to say it's really fun to be a clinician scientist. It's a huge amount of effort, but it's really fun. Like Tina, I not just during the pandemic, but most days, I comment on how wonderful it is to be able to do both of these things. If one of them is not going well, I can sort of fall into the other one, and vice versa. And it's just been a really rewarding career for me to be able to do both of these things.
Absolutely, and I think indeed having the opportunity to do both allows you to tackle some really difficult problems in the lab. Because on the clinical side, we typically get immediate gratification, and thankfully, most of our patients are very grateful because they do well. They can't hear, and now all of a sudden they can hear after you place a cochlear implant in or after you do a stapedectomy. They quite literally think that you walk on water, which is very humbling, but it makes you feel good. So that then allows you to go back to the lab and work on some really difficult problems where you know that 80% of experiments will not work. And you don't even know if the direction is the right direction, but you know that it's important to pursue because it's an unsolved medical problem. So I think the combination of the two leads to an incredibly fulfilling life. It is a very rich life, a very dense life with lots of activity during the day and night, and lots of sleepless nights along the way. But it's really how we spend our energy as opposed to time. I think a lot has been written about time and time management, but I think a more important aspect, and books have been written about that as well, is energy management. And what gives you energy? It's not that everything takes energy away. So when you have these very personal and touching experiences and interactions with your patients, and what your trainees, when their eyes just light up because they now understand what it is that we are working on and why, because they accompanied you in clinic and now they saw a patient who was crying because they couldn't hear, then it all makes it worthwhile and you are energized at the end of it all. Or even if you operate a whole day, sometimes I come home and my husband says, "Wow, that was a long day in the operating room, and are you exhausted?" And I'm not, because I feel energized at the end of it, because I feel like this was worth doing. The patient is better off. It was a good outcome, and I feel like I spent my day well. And the same goes with brands that get rejected. That's very upsetting. It takes a lot of energy. You develop tougher skin along the way. But still, it's very personal. And yet, you know that this is not defining you. It's not a statement about you. You move on, you improve the work, and you keep going. So indeed, it's an incredibly fulfilling career. And there is a psychologist who actually studied this question. His name is Csikszentmihalyi. He was a professor at the University of Chicago, and he studied people in all walks of life, from scientists to physicians to movie stars and construction workers and folks who work on the assembly line and in the entertainment industry, and basically asked the question of what allows people to be in the flow, where flow he defined as optimal experience, where you are fully immersed. And out of all professions that he studied, he concluded you can be in flow in any profession. People on the assembly line can be in flow. They can be really fulfilled. But relatively speaking, which profession allows you to spend most of your time in flow, it's being a clinician scientist. And in particular, a surgeon scientist. And that was quite interesting to read.
Okay, thank you, Dr. Stankovic and Dr. Rubenstein, for taking time out of your busy schedule and sharing your vast experience and knowledge about being a clinician scientist. It was so enlightening, and I'm sure today we audience learned several new things out of this discussion, and we thank you again for being a speaker for this session. Thank you so much.
Have a great
Thank
day.
you.
Have a great day.
Bye-bye.
Okay, bye-bye.